go. It's good to see you all today. We have some Monday students here today and some Thursday or Wednesday. It's not Thursday yet, is it? Some Wednesday students here. So it's great to have you all joining us. All right. Anybody have any questions before we get started? Don't see any hands up. All right, there we go. Let me know if you cannot see where it says cell growth and division there. Good. Always like to double check just to make sure. All right, so last week you guys learned about cell growth and you learned about regulating the cell cycle. So this is all so important that I want to go into a little bit more detail. Now, if there's time at the end, we're also going to talk about Punnett squares, which you're learning about this week. But if not, we will talk about them next week before your exam. Okay? I do have a couple of little cute cartoons, a little cute cartoon here. Today, we're going to learn about mitosis. I need your undivided attention. Undivided. How about this one? Jokes about mitosis aren't funny, especially when they're told twice. Why should you never talk about mitosis at a party? Because it's a divisive topic. And dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. And this is a cute way to remember mitosis. Mitosis. All right, that's it. That's enough of that. All right, now what's really important to remember is that we didn't know anything about cells until the invention of the microscope. That's because cells are too small to be seen with the human eye. The only human cell that we can see without a microscope is the egg cell. A human egg cell is barely big enough to be able to be seen. And of course, then frog eggs and chicken eggs and some other animal eggs are big enough to see. But for the most part, cells are just too small. And there's a pretty good reason for that. There's a good reason that we don't have huge cells. So take a look at these two dogs. They're the same size. Room A has a lot more volume than room B. So volume is the area inside, the space inside the walls. So even though the bigger room also has a larger surface area, walls and floor and ceiling, than the smaller room does, the surface area to volume ratio is a lot less. So kind of a way to remember this is think about if you had to feed that dog and you had to carry away its waste products. It would be a lot easier to get to the dog in the smaller room than it is in the bigger room because it's a long way from the walls to the dog in that big room. It's not very far in the small room. So now when we apply that to a cell, the cell, remember, has a nucleus and it has organelles in it. Now, the cell size is constrained or held from growing to be too big by this surface to volume ratio. If the cell gets big, if it expands in diameter, the volume inside increases more rapidly than the surface area, the outside here. A cell that is too large will not be able to move things in and out of the cell. The smaller the cell is, the more efficient the materials are crossing the surface, right? So what that means is when the cell has too much volume, when it's too big, the nutrients like oxygen might be used up before they get to the middle of the cell. So cells usually don't get too big because they would have trouble moving nutrients and things like oxygen and other molecules from the outside to the whole inside of the cell. You can see that here. So look at these two cubes. These are gelatin cubes that have been put into a substance that the white is the substance, the clear around the outside. It has diffused in. So think about how long it would take something to get from here to the center of this cell compared to this one. It would take a really long time for something to get to the middle of the bigger cell. So smaller cells like this one have a greater surface area to volume ratio. And that makes it easy for the cell to get the food that it needs and to get rid of its waste products. As the cell gets bigger, the volume increases much faster than its surface area. Large cells have a smaller surface area to volume ratio, and that makes it harder for cells to get food and remove waste, right? All right, the membrane now, that's this part around the outside, the orange right there, that represents the surface area of the cell, and the inside of the cell here represents the volume. So you can see it again here. You can see here that when we have a smaller cell, this is a 1 millimeter radius cell, the surface area has a ratio of 4 to 3 or 3.0. 2 millimeter cell, look at the surface area here, and then a bigger cell, even more surface area. Now, they can't get big. Cells just can't keep getting bigger, but organisms do. We do. So a cell has to divide in order to keep the right amount of surface area to volume ratio, in order to keep the right proportion between the membrane and the stuff on the outside. So the solution to this, if cells can't get too big, we have to have cell division. We have to form 2 daughter cells where there was only 1 cell at the beginning. So the daughter cell is going to grow a little bit, and then it's going to divide and be 2 smaller cells, which will then grow and then divide and become 2 more of those daughter cells. All right, now let's look at these words: chromatin, chromatin, chromosome. Yeah. So they all kind of sound the same, but they're all very different from one another. So chromatin is on the left there. It looks like a bowl of spaghetti. It is all loose inside the nucleus when it's not getting ready to divide. So if it's not dividing, it's not called a chromosome. It's called chromatin. When it starts getting ready to divide, it begins to wrap up around these protein balls called histones. It wraps around those and then into these sticks that are called chromatids. Each one of these is called a chromatid. Now here's where it gets really tricky. It's also called a chromosome. 1 is a chromatid, 1 is also a chromosome, and 2 is a chromosome. So sister chromatids that are joined here at the centromere, the mere in the center, the centromere, those are also called chromosomes. I know it gets a little bit tricky. Remember, these sister chromatids are identical to one another. They're called chromatids. They're also called chromosomes. So really, this is a lot like learning a foreign language. It's pretty crazy. Now, what's interesting about this chromatin? Chromatin is a pretty complex molecule. So it's what's called a molecular complex. It's not just one molecule. It's made up of DNA and RNA and proteins, all inside the nucleus of the cell. And it looks like a solid there, doesn't it? When you think of spaghetti, you probably think it's solid. In the pictures that you see, that's how they have to draw it, so that it looks a lot like a bowl of spaghetti, like a solid. But it's actually what's called a condensed dynamic gel-like state. So condensed meaning it's compacted together, dynamic meaning it can change, and it's a gel-like state. It's not exactly solid, not exactly liquid, not exactly gas. It isn't easily classified into one of those states of matter that we think of when we think about things on Earth. Now, this organization, this condensed dynamic gel-like state, allows it to be compacted really tightly together, but still be flexible enough to allow things to happen to it, like repair and transcription and replication. That all can happen because it's able to change. Now, the changeable nature of it is really fascinating, and it's kind of new. We've only really known that it can switch between being sort of more solid and more liquid within the last couple of years, is when we've discovered this. So we know that it can change. It can switch between being what's called heterochromatin and euchromatin, depending on what the cell needs. So that dynamic nature, that ability to change, gives it properties that are sometimes like a fluid and sometimes like a solid at different times, depending on what's going on in the outside of the cell, what other molecules are around it, or in different regions along the DNA. So you could have a section that is more gel-like, and a section that's more liquidy, and a section that's more solid. Now, the most recent studies within even the last year have suggested that it may be capable of what's called phase separation. meaning it can form liquid-like droplets within the nucleus under certain conditions. So it can even go all the way to a liquid under certain conditions. It's a really unique molecular complex, and we're learning more about it all the time. Who knows what we'll find next? Pretty fascinating. All right, here's another cute mitosis cartoon for you. It happens every second. So, hey, yeah. So stop and think for a minute about this. Stop and think that this is happening, this process you've been learning about, interphase, prophase, metaphase, anaphase, telophase, happening in every cell in your body at every minute of the day. We have 172.8 billion cells that are being produced by the human body each day. That's 2 million cells that are being replaced in your body every second. Okay, 2 million, 2 million more, 2 million, 2 million, 2 million. There are 86,400 seconds in a day. When you multiply that times 200 every second, that's 172.8 every day that are being produced. Now, mitosis happens constantly, but the rate at which different kinds of tissue and the cells that are in them will replicate really depends. It depends on the function of the tissue. Can anybody guess where you would find the most rapidly dividing cells? The ones that reproduce themselves a lot. Where in the body could you find those? Around injuries, yeah. Yeah, definitely. Everything kicks up when you find injury. Yep. Cuts, yep. Blood cells. Yep. Which tissues? You know, where, like which tissues in the body? Skin is one. Yep, for sure. Skin is one. Let's look at a couple here. So skin cells. The cells in your outer layer skin are completely replaced every 28 days. So you have a whole new outer layer of skin every 28 days, once a month, basically. So mitosis is constantly happening to regenerate those new skin cells. And when there's damage, as you guys pointed out, then there's even more. There's even more of the mitosis happening to give us new cells. The lining of your intestine. Think about what your intestine is exposed to. You know, basically, your intestine is exposed to the outside environment. You're a tube within a tube. So the inside of your intestines is really outside your body, right? Think about it. So they have a very high turnover rate because they're frequently damaged by the things that we swallow, the things that we eat, and the things that are in the food that we eat. Every couple of days, you have cells that are reproduced in this intestinal lining. Another one here are bone marrow cells. So they are produced very fast, these new cells. Blood is made in the bone marrow. We'll learn that when we get to the anatomy part of our course. But inside your bone marrow is where blood cells are made. Now, blood cells, interestingly, do not replicate themselves. That's pretty interesting. Once a blood cell is, say, made in the bone marrow, very shortly after it's made, it kicks its nucleus out. It ejects its nucleus. So it doesn't have any DNA anymore. It can't do any kind of repairs. It can't make protein. Its whole purpose is to carry red blood cells or to carry oxygen. So those red blood cells get rid of their nucleus. Now, the white blood cells are a little bit different. We'll learn about them when we get to blood cells. But the red blood cells, they only live for about 3 months, about 120 days, 3 to 4 months. And white blood cells actually live less time than that. They live shorter. They are not, well, we still call them cells, Marianne. That's a really good question. They do not become prokaryotic because they still have all the other membrane-bound organelles. Yeah, they still have all the rest of them. So the only ones that are prokaryotes would be the bacteria and the archaea. Those are the only prokaryotes we have in our, in our hierarchy of organisms and cells. Now, constant mitosis is required for the white blood cells, but the red blood cells can't do it at all. But the bone marrow cells that make the red blood cells, they're produced very rapidly because they need to keep up with the supply of red blood cells that they produce. All right, so those are the fastest developing or dividing cells in the body. In the middle, the moderately dividing cells would be ones like liver cells. So they don't divide as often as skin cells do, or as intestinal cells do, or bone marrow, but they're damaged as the liver has like 500 functions that we know of. It's pretty crazy that the liver does. So some of those are, the cells are damaged in the process. So these cells have to undergo mitosis in the moderate range. And then there are some kinds of cells that really don't divide much, if at all. Anybody know what those would be? Muscles and nerves rarely, if ever, divide. Yeah, the heart is one. Heart is muscle. It's cardiac muscle. And it, unfortunately, it'd be nice if it did, because then people who had heart problems would recover much better than they do. So nerve cells. The neurons that you have in your brain and your spinal cord really don't undergo mitosis. Now, once in a while, in the ones that are in the outer part of the body that are not part of the brain or spinal cord, there can be some conditions in which they, yeah, I'll get there, Leo. Hold on just a second. Good question. There are some conditions under which some of the, what we call peripheral nervous system neurons will divide, but most neurons, after they become adult neurons, after they're what's called differentiated, most of the time they just don't recover. That's why a brain or spinal cord injury is permanent. Muscle cells are a little bit different too. You have, pretty much at birth, most of the muscle cells you're going to have the rest of your life. They just get bigger. Pretty interesting, isn't it? Look, when you look at a baby. Skeletal muscle cells don't undergo mitosis. But there are some cells that are called satellite cells that are like stem cells for muscles, and they can divide in order to repair some of the damage that we do. So Leo says, when you exercise, you injure muscles and then you recover again. So you're not destroying the cells the way, like when I scrape my hand on something, I'm destroying those cells. Muscle cells are stressed and they can undergo injury. You can see a muscle cell with, yeah, some of them you can, yeah, some of them, and some of the nerves you can too. They're individual cells that, if you can tease them apart from the muscle, yeah, you can. So these mature skeletal muscle cells, they just don't divide. What happens is when you really work, when you really exercise, what happens is you are strengthening the proteins that are inside the muscle cell. So muscle cells, again, we're getting a little ahead of ourselves here. When we get to muscles, we'll learn that the way muscle cells work is they've got proteins that overlap each other. And when they slide close to each other, the muscle shortens. And when they relax, they go back this way. So the proteins that are involved in that sliding filaments that we have there, those can become, stronger is not exactly the right word, more efficient. They can become more efficient. And muscle cells have a ton of mitochondria. You know what mitochondria do, right? They're the energy producers of the cell. Well, there's hundreds, if not thousands, of mitochondria inside every muscle cell. When you exercise, the DNA inside those muscle cells will produce more mitochondria. It'll give the ribosomes the instructions they need to make the protein to put together those mitochondria. So that's one thing that exercise can really do. And it does just make it bigger. Proteins are the muscles that kind of, it's a good way to think of it. Yeah, proteins, I would say, are the muscles of everything. Proteins do almost all the work in every cell in the body. They're absolutely critical. I know that in every class that I have, there's always somebody who is either vegan or vegetarian. If you are, let me just give my plug for, I'm not saying you can't be, I'm saying you need to work really, really hard. You can't just stop eating meat. You have to work really hard to make sure you get the protein that you need. So if you've decided that you're going to be vegan or you're going to be vegetarian, you have to balance your protein intake so that you get the building blocks that you need. All kinds of problems are going to happen if you don't. So don't just become a junk food vegan where all you're eating is chips and peanuts. Yeah. Yeah. So there's some protein in peanuts, but once again, they're a legume. So you have to balance it. You have to make sure that you're getting all the amino acids that you need. We'll talk a little bit more about that later. All right. So overall, when we look at all the different cells in the body, mitosis is always happening in cells that need a lot of renewal, like the skin cells, like the intestines, like the bone marrow to make blood cells. Other tissues only do it under certain conditions, like when they're growing or they're repairing. And then we've got these some cells that really just don't divide. Once they're damaged or once they're destroyed, like the nerves, they just are not going to, they're not going to recover. All right, now I'm going to go really quickly through a review of the steps of mitosis. You guys have done a really good job, those of you who have gotten there with the mitosis questions and the model that you made of mitosis. But so I'm going to fly through this, but please stop me if there's anything that you don't understand or you want more information. Understanding mitosis is really necessary for understanding genetics, and that's what comes next. We're getting into genetics next. So I need to make sure that you understand this whole cell cycle. Now take a look at all these pictures. These are all images of the cell cycle. They all have an interphase, and the interphase is longer in all of them. So you can see right here, this is the interphase here. The interphase here is purple. The interphase here is light green, or this darker green. There's going to be an interphase here, the G1, S phase, and G2. That's all interphase. And then we've got an interphase right here as well. So they all show the same thing, but they're all different ways of looking at it. Just remember that, you know, that some of them are upside down. Some of them have mitosis up at the top. This one has mitosis at the bottom. Just keep in mind that all the stages are all going to be the same. There's always going to be interphase. There's always going to be prophase. There's always going to be metaphase, anaphase, and telophase. All right. Now, from a bird's eye view, when we look at the whole thing, remember the most important part of this whole cell cycle is the DNA. The cell has to make an identical copy of its DNA. If it didn't, the two new cells wouldn't be identical. And it wouldn't be long before you weren't even a person anymore. You would be some strange collection of cells that were all different from one another. So when the cell splits into two, each of those new cells has to have an identical copy of the DNA, identical to one another and identical to the original cell. But what about all the other organelles? What do you think? When a cell divides, telophase happens, cytokinesis separates it into two different cells. What if there's 5 mitochondria on one side and 400 on the other side, the two cells? You ever think about that? What about ribosomes? What if there aren't any ribosomes in the second cell? All the ribosomes went into one of the daughter cells, and there are none. What do you think? Sherelle?
Well, won't there just be like about an equal by like diffusion, by probability, that there will most probably be like about an equal number on each side?
It's highly unlikely that there would be none of one because there's so many in the cell, but they're not there. The cell doesn't move them. It actively moves the chromosomes. It actively pulls those chromosomes to different sides of the cell, right? When those spindle fibers attach and they actively pull, that doesn't happen. When that cell pinches off in the middle, whatever's on this side goes with this cell, whatever's on this side goes with this one. You're on the right track, Lillian. The organelles themselves don't replicate. The DNA has all the information that it needs in order to make all new organelles. So we don't really worry about the organelles that go to the different sides. The DNA will just make more if it needs it. So you have all this DNA in the nucleus. You duplicate the DNA. The DNA goes into the two daughter cells. If more mitochondria are needed, the DNA just sends the message out through messenger RNA, which we'll learn about more. But that messenger RNA, that will then give the code to the ribosomes to make the proteins needed to make all these other organelles that they need. Okay, so you have all this DNA in the nucleus and you have to duplicate it. That's one of the things that happens in this interphase. We can think of interphase kind of like a waiting room before cell division starts, but it's not a waiting room where they're just sitting around. A lot happens in that waiting room. So the black semicircle here, what you're looking at right here, that shows interphase. Now there are phases of interphase. There's G1, that's growth phase 1, the normal growth phase. In the normal growth phase, the cell is just going about its business. It's making protein, it's getting bigger, it's doing the things that the cell needs to do. Then the S phase is where the DNA is duplicated. So S stands for synthesis. So it is the synthesis phase where DNA is getting bigger. That chromatin is duplicating. It's making a whole nother copy of itself. In G2, this is where now the cell's getting ready to divide. So more organelles are being produced. More proteins, the ones that are needed for cell division, are happening right here. And then at the end of this G2, everything is checked to make sure that everything happened the way it should. So we've got a checkpoint here. That's the G1 checkpoint. We got another checkpoint here. And there's also another checkpoint right here in the middle of metaphase. So at the end of G2, if everything's good, then cell division actually begins. This is mitosis right here. We'll talk a little bit about G0 in just a second. So if everything is good at this G2 checkpoint, and then it goes into prophase. That's what happens there. Now, to remember the order that they go in, try to find something. I always think it's good to try to find a little clue, you know, make up a saying or something. So I just use IPMAT, IPMAT is what I think of, but I've heard this one, my pro met anatella. My is the right here, that's the M phases, or the mitosis phase. Prophase, metaphase, anaphase, telophase. My pro met anatella. I just remember interphase, IPMAT. So whatever you can. Now let's look at the 4 stages of them. Remember, we're looking at the whole cell here. This whole blue is the cell. So right here, we can see some mitochondria. Right here, we can see the rough endoplasmic reticulum that has the ribosomes on it. But we can also see some centrioles starting to form their spindle fibers right here. Now you can think of these like fishing poles, and the spindle fibers are the fishing wire. They're going to grab a hold of the chromosomes because this nuclear membrane is breaking down. So the nucleus is like a vault that the DNA stays in. The DNA is so important that the cell locks it in a vault. When it's time for the cell to make protein, it doesn't send the DNA out, it sends out a copy, messenger RNA. The only time the cell is willing to open the vault, so to speak, is when the cell needs to divide. So we need to get those chromosomes loose into the nucleus or the cytoplasm so that they can be pulled to different sides of the cell by the fishing wires, by those spindle fibers. So we can see that happening here. The nuclear membrane broke down. Those spindle fibers are grabbing on. There's the spindle fibers, these are centrosomes, and they grab a hold of this centromere right in the middle. And as soon as they're attached there, then prophase is over. Now, in metaphase, the spindle fibers, again, the fishing lines, they're going to move the chromosomes to the middle of the cell. Now, keep in mind, they don't just form a line, they form a plate. So they're 3-dimensional. They're all the way, you know, if we could see 3D, we'd see a bunch of them right there. That's called the metaphase plate. And the chromosomes form that plate in the middle of the cell. You can see that happening here. The spindle fibers actually move them right there into that metaphase plate. Then we have anaphase. In anaphase, they're reeling in the fish. So the centromeres there are pulling those, are pulling those spindle fibers so that those chromatids, which are half of the X right here, that's one half of the X-shaped chromosome, and that's the other half, they start moving toward opposite ends of the cell, like the fisherman reeling in his catch of fish. The microtubules on the other side, right here, see these little ones sticking out? They start pushing the cell, so that it's making this oval shape. It's not round anymore. It's oval shape because they're pushing on it. That facilitates the pinching off, the cytokinesis. So there's anaphase being pulled apart. And you can see right there, those other spindles are pushing there. And then finally, the last phase is telophase. In telophase, we've still got one cell, but you can see two sets of DNA in it. So the nuclear envelope begins to form around it again. The chromosomes begin to unwind. So they just look like a bowl of spaghetti again. They're that chromatin again. The nucleolus, which is this little structure inside there that makes ribosomes, that starts to form again. And the cell keeps getting longer. It's continuing to elongate. You can see that happening here. But it's still one cell. Still one cell, but there's two nuclei inside it. Now, at the same time that telophase is happening, cytokinesis is starting. So cytokinesis is where the cell pinches in the middle and before it breaks off. It pinches in at the center at a place called the cleavage furrow. And then you've got a contractile ring of these microfilaments. They pinch the cell in even more until finally the cell splits off into two. And you have two cells where originally you had one. Now, all the organelles start assembling using instructions from the DNA, and the whole thing is done. And now the cell is in interphase again. Soon as cytokinesis is over, it's in interphase and it stays that way. All right, is it always 4 chromosomes that are being divided? No, lots. So we have 46 chromosomes, 23 pairs in people. So whenever any of your cells divide, they are making 46. All different organisms have different numbers of chromosomes. Some have a lot. The only point of DNA. Well, the point of DNA is to hold the code to make protein. That's what DNA does. It holds a code to make protein. Proteins do almost every job in the cell, like way almost every job in the cell. There's very little that's done that doesn't involve protein. So it gives you an idea of how important protein is. Proteins do all the work. The code for making the protein is in the DNA. You can think of the DNA as being the instructions for all the building material that needs to be assembled inside the cell. And that's what DNA is all about. So here we can see the whole thing happening in this little animation. And we're done. 2 cells where we all had one. Now remember, this is all 3-dimensional. So if I move this around, you can see that it is 3-dimensional. We're going to see the metaphase plate here forming. 3 dimensions. Pulling apart. All right, very cool. Remember, once again, this is happening 172.8 billion times in your body every day. All right, really a pretty important process. Very important process. All right, anybody have any questions about that before we talk about this? We saw those checkpoints, right? There are a lot of ways, actually, that the cell controls cell division. How many cells die each day? When you're growing, you're going to have more cells that are being produced than are dying. Once you get to the point that you stop growing, you have a pretty even balance. You've got to have the same number of cells. Otherwise, you'd either grow or shrink, right? You either get bigger or smaller. We don't shrink, but you know what I mean? Yeah. All right. So we've got all these ways, all these checkpoints. There are lots of ways that the cell controls cell division so that these unhealthy cells, any cell that has any kind of damage to the DNA, keeps from duplicating. So if the whole point of DNA is to make protein, think about what happens if DNA is damaged. We don't want a cell with damaged DNA replicating, because it's damaged DNA is going to make the wrong protein or a bad protein or an unfunctioning, dysfunctional protein. So we've got controls to keep that from happening. But of course, sometimes those controls don't work, and the result can be cancer. So I want to look at a little more detail than your book gives you about what cancer is. Just to give you, gosh, appreciation is not the word, but man, are cancer cells a good villain. Let's put it that way. They're a really good villain in the world of villains. Let's watch this video.
What is cancer? Cancer starts in our cells. Cells are tiny building blocks that make up the organs and tissues of our body. Each cell has a control center called a nucleus. The nucleus receives signals from the body to say when the cells should divide and grow, and when they should stop. These molecules can work inside the cells or on their surface. The nucleus then tells the cell what to do. Usually cells divide in a controlled way to make new cells. When a cell is no longer needed or can't be repaired, it gets a signal to stop working and die. Sometimes the signals go wrong and the cell becomes abnormal. The cells keep dividing and making more and more abnormal cells. These cells form a lump called a tumor. Not all lumps are cancer. Doctors can take a small sample of cells from the lump. This is called a biopsy. They will look at the cells under a microscope. This will allow them to examine the cells and decide if the lump is cancer or not.
So basically, the word cancer is a general term for diseases in which cells lose their ability to do all that regulation of the cell cycle. They don't have the ability to stay naturally developing anymore. Now, a lump of cells, any lump of cells is called a tumor. So when you hear the word tumor, it just means lump of cells. Some tumors are not cancer. They're benign. For some reason, they just start growing, but they don't get all of these unique kind of things that we're going to talk about cancer. They're just a benign growth. Some of them, though, are what we call malignant. So malignant ones are cancerous. Sometimes benign cancers can get big enough that they can cause trouble. They can put pressure on whatever's next to them, and they can cause some trouble. Malignant cells, they're the real bad ones. These are the ones that divide uncontrollably, and they destroy the healthy cells that are near them as they crowd them out. And they can move to other parts of the body. They can do what's called metastasizing, where they go to other parts of the body. And as soon as they get to another part of the body, they start growing there. That is metastasis. So what do you guys know that causes cancer? Throw something out there. Yep, radiation, Shmuel, good. Yep, the sun. Too much sun, let's put it that way. Too much of the wrong kind. Reagan.
Smoking.
Yep, smoking. All right, let's look at a couple of them. So cancer is a mutation in the DNA, right? DNA mutations are what cause cancer. Often those mutations are caused by environmental things on the outside. Rudra, alcohol or drugs. Yep, that's a possibility for sure. So exposure to things that are cancer causing can lead to cancer. So certainly tobacco smoke, that's one that we know for sure. Asbestos fibers. They used to use asbestos in building materials. It's not legal to do that now, but a lot of times older homes may still have some asbestos in the building materials that they have, and that gets down into the lungs and can be a carcinogen, which is what we call something that causes cancer. X-rays can cause cancer. The X-rays that they use right now are much, much, much safer than the ones that they used even 50 years ago. But still, too much X-ray radiation can lead to cancer. Too much of the wrong kind of ultraviolet light. So we need some ultraviolet light. We need some skin exposure to the sun in order to make vitamin D. The trick is finding the balance. And we will talk about that a little bit later. Basically, you need to be in the sun almost every day for about half the time it takes you to get a sunburn. So if you've got light skin, like I sunburn in about 20 minutes. I'll start feeling pink. So I need to be in the sun for about 10 a day. If you're someone who has much darker skin and you know that it takes you 2 hours to start feeling a burn, then you probably need to be in the sun for close to an hour every day in order to make the vitamin D that you do. But we don't want to be in the sun too much because too much of it can lead to cancer. Now, really what it comes down to is the sun doesn't cause cancer. Sunburn causes cancer. That burn is what does the damage to the DNA. So be in the sun, but don't get burned. Viruses and bacteria can also lead to cancer. They can be carcinogens. All right, now let's look at how cancer starts. Now, to understand that, we have to go back to the cell cycle. That's why we reviewed it today. Remember that the cell cycle has these checkpoints. This is the G1, the normal growth phase. This is where the cell will check to make sure, are there enough nutrients in the cell for division to occur? Are there enough growth factors? And is the DNA okay? Is it normal? So the cell checks for that at the G1. If in this point, the cell says, wait a minute, things aren't right here. This DNA is damaged and we can't repair it, it will stop growth. It doesn't really send the cell somewhere. What it does is just stop this whole process in its tracks. So the cell won't start duplicating its DNA. It won't go into mitosis. It'll just live out its life, do what it needs to do. And when it dies, it dies. It never makes a daughter cell. There's a condition though. I'll get to that in a minute. There are some times that a cell will just stay in a G0 state for maybe a long, long, long time, and then something will happen and allow it to go back into the cell cycle. So the G0 isn't always a death sentence for a cell. All right, then we've got another checkpoint right here. The G2 checkpoint makes sure the cell's the right size, and it makes sure that all the DNA was synthesized properly. And then right here in metaphase, they make sure that the spindles are attached correctly to that spot in the center. If those spindles are not, then the cell will not continue on. Now, these checkpoints look for problems. If they find it, they fix it. If they can't find or they can't fix it, the cell either goes into G0 or it commits cellular suicide. We call that apoptosis, where the lysosomes, remember what lysosomes are? They hold those digestive enzymes. The lysosomes will break open and destroy the cell itself. That's how important it is to the body to keep from getting cancer. It will kill its own cells in order to keep them from dividing in the wrong way, uncontrollably. Now, in normal cells, when a normal cell comes into contact with the cell next to it, it stops growing. There are signals, they're called anti-growth signals, and they stop the cells from continuing to grow and crowd each other out. There are also things that are called tumor suppressor genes right in your DNA. These tumor suppressor genes keep cells from growing out of control. In cancer, that all changes. The cell stops going through the normal cell cycle. Mutations in the DNA are not caught and fixed. And those mutations often affect the genes that are part of the checkpoints. So cancer tricks the checkpoints into letting the cell continue to divide. So the cells that have these mutations in their DNA, they keep going through the cell cycle because there's nothing there to stop them. Checkpoints aren't working the way they're supposed to. The tumor suppressor genes aren't working the way they're supposed to. The signals that you're getting too close to other cells, they're not working the way they're supposed to. So these cancer cells just grow and divide. And as they grow and divide, they build up more and more and more mutations. They actually even will stop the genes that cause apoptosis. So the tumor suppressor genes aren't working. The anti-growth signals aren't working. The apoptosis message isn't working. They just are growing and crowding and growing and crowding and continuing to cause trouble as this all happens. Now, the genes that usually guide proper development, they stop working too. So the ones that stop improper development, they're turned off. The ones that guide proper development, they're turned off. So once again, we have these abnormal cells that divide and divide with nothing to stop them. They form this irregular lump that doesn't have any kind of organized arrangement. This is what a lump of cancer cells look like. Remember that this is what normal cells are going to look like. Very, very structured, very orderly. Let's see here. Okay. So even the cells themselves look different. Marianne, sort of like overpopulation. Yeah, exactly. You got too many cells in a small space that they're not supposed to be there. And the cells themselves look different. Their inner membranes are faulty. Their inner structures are faulty. Their nuclei are big and they're not regularly shaped. Look at the shape of the nuclei in a normal cell. Now compare that to a cancer cell. A normal cell, you're going to have these dividing cells that are orderly arranged, not so in a cancer cell. Normal cells have a particular shape. They lose that. They gain a lot of variation, so they're not like each other, and the features are lost. In this one, you can see the nucleus. You can see, the different structures. You lose all that. So these changes are going to disrupt the function of the cell. The cell isn't going to work the way it's supposed to. I think this is one of the kind of interesting things, most interesting things about cancer cells is that they can grow their own blood supply. Watch this video and see that.
Cancer starts when cells multiply out of control, forming a tumour. Once the tumour reaches a certain size, about the size of a grain of sugar, it needs its own blood supply. The tumour produces chemical signals which reach the blood vessels nearby. These signals hijack the blood vessel, causing new vessels to sprout and grow towards the tumour. With a blood supply in place, the tumour is provided with oxygen and nutrients so it grows. Because the tumour is now connected to the bloodstream, cancer cells can break away and travel around the body. This is one way that cancer can spread.
So this process is called angiogenesis. Angio refers to blood vessels. Genesis means to make. So it's sending these messages. And think about, did you hear the size of that? When it's about the size of a grain of sugar, that's pretty tiny. Then it starts sending these messages to the blood vessels that are by saying, I need some blood. I need you to grow toward me. So in a nutshell now, cancer cells stop going through their normal cell cycle. They usually contain several different DNA mutations, and those mutations often affect the genes that are involved with the checkpoints. The genes for apoptosis or programmed cell death are usually suppressed. Genes that maintain proper development stop working. The signals to stop growing when they contact neighboring cells stop working. The cells grow and divide uncontrollably. They can grow their own blood supply. They can evade immune system detection. They often reduce the expression of molecules that allow the immune system to recognize them. So they kind of like think of it that they camouflage themselves so that your immune system doesn't recognize them. They can evolve mechanisms to repair their damage to themselves. So they actually have been shown to, so one way that they will treat cancer is by giving you medication. Chemotherapy is medication. These cancer cells evolve ways to pump those drugs out of the cell. Think about that. They repair DNA damage that's caused by the treatments. So radiation and chemotherapy will damage the DNA of these cancer cells. They have ways to fix that. And they can undergo mutations that make them less susceptible to the therapy that we're trying to do. It's pretty crazy. Whoop, went too far. Here we go. And they can spread to other parts of the body. So that's one of the biggest problems here. That's called metastasis. And that's the ability of these cancerous cells to move to other parts of the body. Once they get there, cancerous growths can form. So the primary cancer is where it starts. Where it metastasizes to often determines how deadly it is. So if something metastasizes to the lungs, you're going to get cancer in the lungs. Cancer in the lungs is not usually a good thing to get. Cancer of the brain. I mean, cancer anywhere isn't great, but there are some that are way more deadly than others. Nice try. Yeah, exactly. Nice try. Yeah, it's pretty crazy. So how do we treat it? Basically, once a person has cancer, you have two options. You either take it out or you kill it while it's still inside. So surgery is a way of taking it out. They cut it out. They quite literally go in, identify where the cancerous cells are, cut them out, and remove them from the body. Often that's not enough, especially if a cancer has metastasized. If a cancer is moved to 5 parts of the body, you can't really do 5 surgeries to get it all out. So a lot of times they'll use surgery in conjunction with chemotherapy. Chemo means chemical. So chemotherapy uses chemicals to try to kill the cancer cells while they're in there. And one thing, cancer cells have a couple things about them that make them pretty good targets for these treatments. Because they divide so fast, they've given chemicals to try to slow down the cell cycle, slow down mitosis. There also are some proteins that are specifically found in cancer cells. So if you can find a way to destroy those proteins that are only in cancer cells, then you can get rid of the cancer without doing a lot of damage to the healthy cells. But there's always some damage. If you know someone who has had cancer treatment, either chemotherapy or radiation or surgery, there's always some kind of damage done to the healthy cells. So the radiotherapy, sometimes this is called radiation. This uses very high energy light rays in order to destroy the cancer cells. So they can very, very specifically pinpoint right where those cancerous cells are. Now, there's a thing called immunotherapy that they're really starting to work with a lot. It's not tremendously effective right now, but it's being explored and they're hoping to improve it. So this uses the body's own immune cells. It takes them out, takes out some immune cells, some white blood cells basically, and then it changes them in some way, and then it puts them back in the hopes that they will now go after those cancer cells. That, in other words, the cancer cells won't be able to be camouflaged from them anymore. It helps them. For breast cancer, the person loses hair. For some, that's true of a lot of cancers, Marianne. Yeah, very good point. For some reason, the hair follicles seem to be especially damaged by the chemotherapy drugs that they use. Yeah, yeah, pretty interesting. So yeah, I mean, I think there's a lot of promise with this cancer immunotherapy. There aren't a lot of side effects because you're putting the person's own immune system cells back in, and there isn't a lot of damage done to the healthy cells because it's only the immune system. So on every one of your cells in your body, you have markers that say, I belong to me. You know, Reagan, your cells all have a marker that's very different than Umar's, which is very different than Marianne's. So if I took some cells from you and put them in Marianne, your own immune system cells would say, wait a minute, those don't belong here, and it would take them out. Well, since these immunotherapies would be your own immune system cells, they're going to recognize your own body, and they're not going to attack your body. They're just going to attack the cancer cells that are very different. Isn't that the problem with organ transplants? What the problem?
The fact that each person has their own individual marker.
Yeah. So a person who has an organ transplant has to take an immune suppressant medication for the rest of their lives. Yep. They have to turn down their own immune system. Could you create a cell that's malignant that only penetrates cancer cells but not body cells? You work on that, Leo. You'll make enough money to take care of all of us for the rest of their lives. That would be really nice if we could create a cell that only or anything. You know what's interesting though? So we hear a lot about cancer. If we were to eradicate cancer, if cancer was gone from the face of the earth, it would only increase the lifespan of the average human about 5 years. Think about that. Cancer is not the biggest thing. There are a lot of other conditions that can do a lot of damage, that can kill people. So cancer definitely is something that we want to work on not getting. As I told you, all the things that cancer cells do, the best thing to do is just not get cancer in the first place, right? So there are a lot of things that you can do. All those things that I said cause cancer, avoid them. Avoid toxins that are likely to be carcinogens. The most dangerous thing that kills most people, that changes, Leo. That changes from time to time. I'm gonna look it up. What is the number one? Number one cause of death for humans, cardiovascular disease, heart disease. So that includes strokes, heart failure, heart attacks, and other heart-related things. So listen to the major risk factors for cardiovascular disease, okay? And think about whether or not you have any control over any of these. High cholesterol levels, smoking, diabetes, obesity, a sedentary lifestyle, a diet high in salt, sugar, and unhealthy fats, and alcohol consumption. That's what the risk factors are for heart disease. So what can you do? Avoid those risk factors. Yeah, I know someone who has cancer. As far as I know, they've been really healthy and have gone to the gym very frequently. Yeah, so one of our big mysteries is why some people seem to live really unhealthy lifestyles and they live to be 90 and perfectly healthy, and others seem like they're doing everything right. A lot of it is genetics. A lot of it is factors that are beyond our control. Like, you know, I can choose not to smoke. I can choose to eat a healthy diet that means that I'm not going to have diabetes. I can do all that, but I'm not really in control of the air that I'm breathing. I can't really change that. I'm not in control of the whatever would be in the water supply of the water that I'm drinking. I'm not in control of any of the environmental factors that play a role. I can control some of it. Others are harder. Yeah, others are harder. Absolutely. Absolutely. But it's the secret. And as we learn about more and more about living things, and especially when we're talking about people, both in this next couple of lessons, a couple of chapters, and then in the human anatomy part at the end, we're going to learn that it's just way easier to stay healthy than it is to get healthy later. So do the best that you can now. Make wise choices. That's the best way to put it. All right. And with that, we are out of time. Anybody have any questions? We did not get to Punnett squares, so we will talk about those next week. All right. Have a great week, everybody.
Thank you.
Thank you. Bye.
You're welcome.